deals with plasmids. Okay. What we know about plasmids is they are circular, right? They contain anywhere from 5 to 100 genes. They are separate from major chromosome, right? So they're extra chromosomal. And they replicate on their own, meaning that when the cell goes through cell division, replicates on its own, the plasmid replicates on its own. So that means that if this cell has a plasmid in it, when that cell divides into another cell, of course it's going to replicate its DNA, so it has a copy of the DNA in each one, right? Same thing with the plasmid. The plasmid will replicate on its own, so each cell will have an exact copy of the plasmid, okay? So it does replicate on its own. Now, the characteristics of it as far as what it is able to do. It's able to code for auxiliary functions. And examples of those auxiliary functions are, morning. Examples of those auxiliary functions are, it could code for antibiotic resistance. It could code for special toxins that it could put out. Now, these toxins could be either to the host, or it could be a toxin to another microorganism, such as we have a gut bacteria, right? And this gut bacteria will put out toxins to kill off any other bacteria in the area so that only it can grow, right? So it's both used as a defensive mechanism as well as a mechanism to poison the host, basically. It doesn't know it's poisoning the host, but it's putting this out and it's going to harm the host. So yeah, antibiotic resistance, toxin production is also one of these things that it can produce. And when we get into microbial genetics, we'll talk about how this transfer actually happens. It actually happens through what we call conjugation. And we'll go into that later, but don't worry about that. So as long as you have all that within that essay, you should be good as far as explaining that question.
Okay, for example, like I have circular with 5 to 100 genes, replicates, produces toxins, and is antibiotic resistant. Would that cover everything? Would you have to do that one?
Okay, but are you going to give an example of some of the auxiliary functions, such as
Produces toxins,
toxins
and is antibiotic resistant.
and can code for antibiotic resistance?
Okay, so I need to put can code for toxins and code for.
Yes.
Okay, would be better. That's what I want. Code for toxins and code for antibiotic resistant, right?
Yes. Okay. Next question is, describe what glycocalyces are and describe the two types of glycocalyces we talked about. Be specific in the characteristics of each type, meaning that each type has special characteristics that make them different. So basically, what is a glycocalysis?
Gelatin-like sticky layer.
On the external,
Periodic and eukaryotic.
yeah, it's on the external surface of the cell, right?
Could it also be protects the cell from drying? It can play a role in the ability of pathogens to survive and cause disease?
Yeah. And when we talk about the specific, ones,
Yeah.
like the capsule or the slime layer, okay? Now your capsule, its characteristic is it is tightly bound to the cell, right? It's well organized in structure. It has the ability to attach to cells, right?
Yeah.
The capsule also protects it from what? Drying out
They're becoming phagocytes.
and becoming phagocytes. So your immune cells are going to try and phagocyte them, bring them in and destroy them. Well, that capsule is going to protect them from being phagocyzed. So once they get put inside the cell, they will not be able to be phagocyzed, but they will be able to continue to replicate, okay? That's part of its virulence factor, meaning that virulence means the ability to cause disease, okay? Now, if we're talking about the slime layer, characteristics of the slime layer, it is loosely organized, right? It is water soluble, right? And that characteristic itself gives it the ability to form biofilms. On our teeth, in the water. My example I gave you was Vibrio cholera, because it has the ability to form biofilms in the water, so that when it keeps on proliferating and growing, gets into our water system, we drink the water, we get sick with cholera. Okay, what else about slime layer?
I just put that as loosely attached and promotes biofilm growth such as black on our teeth.
Okay, and you also want to say that it is water soluble as well. Because if it wasn't, then it wouldn't be able to form biofilms within the liquid environment. Okay, so describe what selective toxicity is, and then how can we use this knowledge to target and help fight bacterial infections. So what is the basis of selective toxicity?
Using an antimicrobial agent that will act upon the bacteria without harming the host.
Yes, that's the most important thing. This is something that's going to harm the bacteria or kill the bacteria without either harming the host or doing less harm to the host. Because some antibiotics, as you know, we have side effects that do kind of harm us, but it's not going to harm us to the point where we're going to get more sick or die, right? And so that's the characteristics of selective toxicity. So how can we use this knowledge in order to target and help fight bacterial infections? And for example, you have to know what the three targets are, right? What are the three targets?
Let's see, when targeting the cell wall, DNA, RNA, protein, and enzymes.
Yes. So we can target the cell wall or cell wall synthesis. We can target DNA replication or RNA synthesis. We can target protein synthesis. And we can target enzyme production. If we can stop protein production, we can stop enzyme production, right? Because enzymes are made up of proteins and other things, right? So, for example, targeting the cell wall, which antibiotic did I state would target the cell wall synthesis?
Penicillin?
Penicillin, because penicillin attacks the enzyme that builds the cross bridges that hold the cell wall together. If I can target that enzyme, block it from working, or basically through competitive inhibition, then it will not be able to build those cross bridges. It's the same thing as if I am building a bridge from one end of the river to the other end of the river, and people are building it, but the thing is, I don't supply them with the steel they need to make it solid together. So now we can put all the cement we want up there, but it's not gonna be held together because that steel is not there to hold it up. So it will just collapse and fall, right? Same thing with the cell wall. If those cross bridges are not made, then that cell wall will collapse, create a hole. And this is going on all around the cell, so all these holes are being punched into the cell, and the cell will eventually burst, right? And we know that penicillin only acts upon what? Actively dividing bacteria.
Divides it?
No, bacteria that are actively dividing, penicillin will only act upon it. If the cell is not going through replication, penicillin is not gonna work against it, okay? What's the other example I gave? Sulfa drugs. Okay, sulfa drugs prevent the conversion of PABA to folic acid. Folic acid is needed for DNA replication. So that's a drug that is targeting DNA, right?
It prevents the production
Of DNA, or it prevents the replication of DNA. Bacteria already has its DNA, but if it wants to divide and make more copies of itself, it has to replicate its DNA. And in order to do that, PABA has to be converted to folic acid in order for this to happen. If I prevent that, then it cannot replicate. If it cannot replicate, then the cell is going to die. Okay? And that's pretty much about it. As long as you mention that those three targets and give at least one example of how we target it, that should be good.
So, we need to list like the penicillin and the sulfa drugs, or can we just put it target 70s ribosomes while not hurting us because we have 80s ribosomes?
You can
as
do
an
that one as well,
example?
because that's another example I gave, targeting the 70S ribosomes. As long as you remember that to tell me bacteria have 70S ribosomes and humans have 80S ribosomes. That's what makes it more specific. Morning, sunshine. Huh? 10 o'clock? We didn't think people actually came?
Well, what time is it? You said 11, didn't you?
It's 10.
Oh my
It's
God,
always
did you
10.
really? Is it 10 or is it 11? Oh, I'm tripping, sorry.
Why do you have it?
Your way sounded
Were you
easier,
up
but I already
partying
kind of
last
studied
night?
it. This
No,
one
I was
was
yeah,
gonna stick with it.
Huh?
Yeah, I was
break it
put
down.
Went to the club?
Yeah.
So the X means that you can drink or cannot drink.
Can't.
So they let you in even if you're underage?
Yeah, like you can get in if you're 18.
What club is this?
Like, it's by 6 Flags. It's called, Spell. No?
Do they play any good music there?
Sometimes.
Does all music have that?
Dun, dun.
Well, no, just
Gotta
club,
have that
club
bass.
music. Club music has that.
Rave flashing lights.
That's what I grew up.
Well, back when I was growing up, we called it disco.
There you go.
Stay alive.
And that began when I was in 6th grade, and none of you guys were even thought of. Do you have a copy of the study guide?
Yeah.
Yeah.
Somewhere.
We are actually on number 3. We're finishing up number 3. But I got through that